And we're going over details of karyotypes and chromosome aberrations. What happens when children or offspring don't have the right amount of karyotypes. Some of you went over this in regular biology. I know you went over it if you were in my class, and some of you were. If we were in class, we would be putting together physical karyotypes, but obviously we can't do that at the moment. But the human chromosome set. The number and appearance of chromosomes in the nucleus of an organism is unique for each species. Since this is a class about human biology, we mainly focus on our set of chromosomes. We have 23 pairs, which means we have 46 total. 22 pairs of autosomes that control the majority of our characteristics, and then 1 pair of sex chromosomes that determines our gender. That's what chromosomes look like. That should be a review. They're banded. The different bands on them are the different genes. You put 2 of them together, you have sister chromatids. You have the centromere in the middle. Keeping them together, that's all the information you should have. So now we're going to go a little bit further and you're going to learn more about chromosomes. There's a long arm and a short arm, and they look different ways. Some of them are longer, some of them are shorter, and they have different names. Telomeres on the end of each chromosomal arm prevent chromosomes from sticking together. They also shorten after cell division, and they're related to aging. A lot of companies right now are focusing on the telomeres of chromosomes and keeping those healthy because they think it's related to aging. They think they can make some anti-aging serums or potions or chemicals or whatever to help people not age so quickly. Chromosomes are identified by size, centromere location, and banding pattern. Like I said, it's not just a chromosome anymore. You gotta learn the different pieces of them. You got a short arm and a long arm. When they're even, they're called metacentric. Meta means middle, and the centric means center, so the centromere is in the middle. Submetacentric is when the short arm, or the P arm, is shorter than the Q, or the long arm. And then acrocentric is when the P, the short arm on top, are just kind of little blobs. And then the Q is the longer arm, and it's still longer. And you can see they each have a unique banding pattern of purple and blue throughout them. Autosomes are all chromosomes other than the sex chromosomes. And they're involved in all of our characteristics except for sex determination. Females have homologous sex chromosomes. We have X-X's. We've talked about this already. This should be a review. Males have non-homologous chromosomes. They have an X and a Y. And they differ in size and shape. Stains and dyes are used to produce chromosome bands. Each arm is subdivided into numbered regions from the centromere to the telomere, And that is the standardized banding pattern for the human chromosome set. Those are obviously drawings. Our real chromosomes don't look like that. But each one has a unique banding pattern. It's like a fingerprint. All of your chromosomes hold different genes and different patterns. Within each region, bands are identified by number. So any region on a chromosome can be identified by a descriptive address, such as 1q 2.4. The chromosome number is 1, the arm is the Q arm, or the long arm, the region is 2, and then the band is 4. So all of the chromosomes are split up in this same way, so you can identify the different regions. So when you talk about genes, you can give it a specific location, or scientists can give it a specific location. An image of a complete set of chromosomes looks like a splat on the bottom on the left. It gets photographed from metaphase because that's when the chromosomes are the thickest. Karyotypes can be constructed from several cell types, including white blood cells, skin cells, amniotic fluid cells, and chorionic villus cells. And chromosomes are arranged in pairs according to size and centromere location. So you get a picture that looks like a splat, and then you kind of have to cut them out and match them up and make sure you have the correct amount. Analyzing karyotypes. Stains and dyes produce banding patterns specific to each chromosome. Like I said, you cut them out of the splat, you match them, you put them together in a chart, and then you can see the number of chromosomes present, the sex chromosomes, whether it's a boy or a girl, and then the presence of absent or individual chromosomes, presence or absence of individual chromosomes. All humans should have 46. If it comes out that somebody gets genetic counseling, they have a karyotype done, and the child has more than 46 or less than 46, then they have some kind of syndrome. We'll talk about the different syndromes here throughout the presentation. New techniques using fluorescent dyes generate unique patterns for each chromosome. Chromosome painting can detect translocations, duplications, or deletions. So it's not just about matching the chromosomes. The chromosomes have to be exact. If one chromosome is matching a certain section, that section has been deleted. Depending on how important that section is, the child can have some defects or syndromes when they come out. Prenatal or fetal chromosome analysis. These happen while the child is still in the mother. A test called an amniocentesis. It's a recovery of fetal cells from the amniotic fluid, followed by the growth of cells in the laboratory for karyotype preparation. It's performed in the 16th week of pregnancy. There are risks of miscarriage. So this is restricted to certain circumstances. This isn't offered to every pregnant woman. Down syndrome is related to the mother's age, so pregnant women over the age of 35 are actually offered this test routinely. If there's a history of chromosomal disorders in the family, then they'll also offer it. But this is not a mandatory test for every pregnant woman. Chorionic villus sampling. Recovery of cells from a placenta, followed by the growth of cells in the laboratory for karyotype preparation. This can be done earlier, in the 8th or 9th week of pregnancy. Again, this isn't mandatory either. But if a woman is anxious, she's pregnant, maybe she's had some miscarriages, maybe she has some chromosomal disorders that run in her family and she wants to know earlier if there's anything wrong with her baby, this is a test that she can request. There's also videos, if you guys want to watch them, that go with the amniocentesis and the chorionic villus sampling to see how they're done. Amniocentesis, there's a big needle. You stick it in the mother's abdomen to get the amniotic fluid. There's also non-invasive methods. They're not as accurate, but they're getting more accurate because technology gets better and better every year. So analysis of fetal cells isolated from maternal blood, so they can take the mom's blood and get fetal cells from the mom's blood. And the tests become more effective every year, but they're not yet as accurate as the other two. Changes in chromosome number can cause genetic disorders. Normally, we are called euploid. You say the EU, like Europe, euploid. That's the normal number of cells. Our body cells are diploid. It has the full 46 chromosomes. Our sex cells are haploid. Those chromosomes have been cut in half to get passed on to the next generation. Those are our sex cells. They only have 23. And there's a condition called polyploidy, which is an increase in the number of chromosome sets. So in the picture, if the haploid number is 3, the diploid number is 6. If the offspring turns out to have polyploidy, they have multiple sets. This is rare. Aneuploidy is changes in chromosome numbers involving less than a whole chromosome set. This is more common. Often it's the gain or loss of a single chromosome. It can either be monosomy, which when you're missing one chromosome, mono means 1, or trisomy, tri means 3. So you gained an extra chromosome, so you have a set of 3 somewhere in your karyotype. Aneuploidy is most often due to a process called non-disjunction. This happens during meiosis. Remember, there's 2 rounds of meiosis. It can happen during either round. Autosomal monosomy, and one of our autosomes, chromosomes 1 through 22, is missing chromosomes in the autosome set. It's rarely observed in miscarriages or live births. It's most likely lethal before the pregnancy is even recognized. So you can see in that karyotype, in the red circle, there's only 1 chromosome there. So there's just missing 1 chromosome. And often that is enough to miscarry a baby. Trisomy, autosomal trisomy. Accounts for 50% of the chromosomal abnormalities observed in miscarriages. Only a few autosomal trisomies result in a live birth. 8, 13, 18, and 21 will result in a live birth. And trisomy 21 is the only one that allows survival into adulthood, and that's Down syndrome. So if a child has 3 copies of chromosome number 21, that child has Down syndrome. If they have 3 copies of any of the other ones, they normally don't last into adulthood. Non-disjunction, like I said, is the process that leads to either extra or missing chromosomes. It can happen in round 1, where you can see the cell splitting, but it doesn't split correctly. It doesn't split the information in half. You're supposed to end up with 2 and 2 on each side. So when it goes into meiosis 2 and then it ends, you have 4 gametes with an odd amount of information. The top 2, if any one of these eggs get fertilized or sperm fertilize the eggs, that child will have an extra chromosome. If any one of these 2 on the bottom get fertilized, that child will have a missing chromosome. And then, like I said, it can also happen in round 2. So round 1 had normal cell division, where the information was split evenly. But something happened in round 2, where the top one didn't split evenly. So this person has a 25% chance of having an abnormal baby, or 25% for extra, 25% for missing. But they also have a 50% chance of having a normal child. Patau syndrome is trisomy 13 when they have an extra number 13 on their karyotype. It happens in 1 in every 10,000 births. It's not very common. Defects in many organ systems suggest developmental abnormalities begin very early on. Parental age is the only known risk factor. And it's lethal, often within the first month of age. So these babies are born. They normally don't survive past 1 month old. Trisomy 18 is called Edward syndrome. This happens in 1 out of 11,000 births, and 80% are females. This occurs more in females than males. Again, parental age is a common risk factor, age of the parents. And it's lethal within 2 to 4 months of age. So the babies get born, but they don't normally last past 4 months of age. And then trisomy 21 is Down syndrome. They have an extra chromosome number 21. It happens in 1 in every 800 live births. This is very common. It's the leading cause of childhood intellectual disability and heart defects. Something about this affects their heart. And despite susceptibility to many health problems, individuals with Down syndrome can live productive lives and may survive up to 50 years of age. And since they are born with heart problems, it's usually their heart that gives out. What are the risks? Well, the exact cause of autosomal trisomy is unknown. Many factors may be involved. Some families have a genetic predisposition, it's just in their genes to be passed down. Exposure to radiation can cause it. Viral infection can cause it sometimes if there's a viral infection in the mother. And abnormal hormone levels. All of our hormones have to be just right within certain levels for everything, for every one of our bodily functions. If they're abnormal, especially in the mother, when she's trying to get pregnant or in early stages of pregnancy, that can cause the trisomy. Advanced maternal age is a major risk factor, especially for Down syndrome. 94% of trisomy 21 non-disjunctions occur in the mother, most likely in meiosis 1. And you can see from the graphs, the risk for Down syndrome increases dramatically as a mother ages, as a woman ages. Like I said, after the age of 35, most women get an amniocentesis or the chorionic villus sampling to make sure everything's okay with their baby. Why is maternal age a risk factor? It has to do with how we make our eggs. So primary oocytes, which are our eggs, develop during embryonic development and pauses in prophase 1 of meiosis at birth. So us women, we have all the eggs we're ever gonna have, we develop them in utero while we're still embryos in our mother. And these eggs pause during prophase of meiosis 1. And the chance for intracellular or environmental factors to cause damage to these eggs increases the longer they're paused at this state. So as a woman ages and these eggs get older, more likely that intracellular, which just means in your cells, or environmental factors will cause damage. Maternal selection. Chromosomally abnormal embryos are typically prevented from implanting in the uterus. So even if it gets fertilized, it doesn't always get implanted. There's so many steps to having a baby and conceiving a baby. Our body usually prevents this for us. And this maternal selection may become less effective as women age, leading to more aneuploid births in older women, more births with extra or missing chromosomes. So when women are younger, our bodies do this for us. It won't implant embryos that have something wrong with them. But as women age, that particular skill goes away or becomes less effective. Sex chromosome aneuploidy, when there's not enough or too many sex chromosomes. This happens. It's actually more common than in the autosomes. It can involve both X and Y chromosomes. At least one copy of an X chromosome is needed for human survival. So if an embryo does not have at least one X chromosome, it doesn't become an embryo, it miscarries. And then additional copies of the X or Y chromosome interfere with normal development. Turner syndrome is one of those. It's monosomy, so there's only one of the X chromosomes. It's the only viable monosomy in humans. This is the only time that a baby will be born with one less chromosome. Happens in 1 in 10,000 female births. This only happens to females. If you only have an X chromosome, you are a female. Boys have to have a Y. We actually have a couple students. I had a student that graduated, I think she graduated in 2014. She had Turner syndrome, and she comes back and talks to my classes every once in a while. And then I know there's another student at the school right now with Turner syndrome, because they met last year on the day that she came to speak. Turner syndrome results from non-disjunction during either mitosis or meiosis. Can happen in either. There's no association with maternal age. Individuals are sterile, meaning they can't have babies of their own. They display some unique physical characteristics, but have no intellectual disabilities. Mostly they're just shorter than everyone else. Like I said, they can't have babies of their own, but there's nothing wrong with their brain. They're just as smart as everyone else. Klinefelter syndrome. This is XXY. It happens 1 in every 1,000 male births. Boys have Ys, so everyone with Klinefelter syndrome is a boy. So Turner syndrome is only in girls, Klinefelter syndrome is only in boys. Both maternal and paternal age are risk factors. And it's characterized by some fertility problems, but few additional symptoms. So to look at these people, you can't see anything differently. They might have fertility problems when they grow up and try to have children of their own, but that's it. Structural changes. Chromosome breaks can occur spontaneously through errors in replication or recombination. Environmental agents such as UV light, radiation, viruses, and chemicals can also produce these breaks. Chromosomes are sensitive to these things. Our bodies are pretty good at getting rid of them if something is wrong, but that is not always the case. Deletion of a chromosome region causes developmental abnormalities, and then deletion of an entire autosome is lethal. Like I said, if you don't have 22 autosomes, 22 pairs of autosomes, something gets deleted, that can cause major problems. There's some examples of chromosome deletions. Prader-Willi syndrome is one that you hear about a lot. Infants are weak, they don't grow as quickly. In children and adults, they're more likely to be obese and have compulsive eating disorders. And then on the left of the table, the deletion is the arm. So the first one happens in the 5p arm, or the p arm, region 5. That's what the numbers stand for. Duplication is a repeated chromosome segment. So you can see in the picture that DE segment is repeated twice in the bottom chromosome. Translocation is when a part of a chromosome is moved to another non-homologous chromosome. There's two different types. There's reciprocal translocation. Parts of two chromosomes break off and just attach to different ones. And then there's something called Robertsonian translocation. You got two pairs of normal chromosomes, and then whole chromosomes become attached to each other. Inversion is when the order of chromosome segments is reversed. And then each of these types of structural changes cause different syndromes and disorders and abnormalities. It's a miracle with all these. Our bodies get it right most of the time. And then some consequences of aneuploidy, besides all the syndromes. Major cause of miscarriages and birth defects. Only 13, 18, and 21 occur with any frequency in live births. The rest are eliminated by miscarriages as the pregnancy progresses. It's also associated with many cancers, especially leukemia. Other forms. In some cases, the karyotype and individual chromosomes appear to be normal, but the phenotype is abnormal. So scientists have to carefully analyze the karyotypes to reveal subtle chromosome changes, like the inversion or the duplication. Those are going to be very, very subtle changes. Other situations include uniparental disomy, which we'll talk about in a minute, copy number variants, and then fragile sites are another rare form of chromosome abnormality, which can be observed only when cells are grown in the laboratory and certain chemicals are added to a growth medium. So you really have to be looking for something to be wrong to detect the fragile site. Uniparental disomy, or UPD. It's a condition with when both copies of a chromosome are inherited from a single parent. Normally, you inherit one from mom and one from dad. UPD of the long arm of chromosome 15. If both copies are inherited from the mother, the result is Prader-Willi syndrome. If both copies are inherited from the father, the result is something called Angelman syndrome. Angelman syndrome. Copy number variants are defined as a deletion or duplication of DNA segments from about 1,000 base pairs, which is 1 kilobase, to about 5 million base pairs, called 5 megabases. CNVs change the structure of the genome, and along with submicroscopic inversions and translocations, they're all forms of genome structural variation. So this is when it happens on a large scale. There are very common diseases associated with copy number variants, Alzheimer's being one of them. HIV, if you have a certain gene, you have an increased susceptibility to HIV or AIDS. Lupus, Parkinson's disease. And fragile sites appear as gaps or breaks in a chromosome. They're inherited as codominant traits. And the breaks produce chromosome fragments, deletions, or other aberrations. Example, the Frax A site near the tip of the long arm of the X chromosome is associated with an X-linked form of intellectual disability known as Martin-Bell syndrome or Fragile X syndrome. And that is all for the notes today. Anyone have any questions? Give me a yes or no in the chat box. Let me know if you're listening. No. All right. Giovanni, stick around for a minute, and everyone else, you are dismissed. Go get to work on some of your weekly work. You guys are watching Gattaca this week and doing a discussion. That's all I have for you guys.
Bye, Miss Maurice. Take care.
Bye, take care.